Hello, this is talk number 18 in the series from the collegiate textbook Principles of Biochemistry, 7th edition, written originally by Albert Lehninger. Today we're going to continue our discussion of lipids. Today we'll be discussing fat-soluble vitamins and address some early components of membrane transport. So the fat-soluble vitamins, there are 4 of them, vitamins A, D, E, and K, and all these must be obtained from the diet. We don't make those. Vitamin D3 is cholecalciferol, is formed in skin by ultraviolet light and converted by the liver to the metabolically active 1,25-dihydroxyvitamin D3. Calciferol is which is this active entity regulates intestinal uptake of calcium and calcium in kidney and bone. The food additive that you have in your diet is vitamin D2, which is ergocalciferol, and is formed by ultraviolet irradiation of yeast, similar in mechanism to that of vitamin D3, which is also metabolically converted. So we get our dietary form is vitamin D2, and it is transformed into the active component. This is a drawing of the reaction process that we have. The 7-dehydro cholesterol is here, and there is ultraviolet light, which breaks the 9-10 linkage right here. Here's 9 and 10 is over there. This breaks this open. This is vitamin D3, cholecalciferol, and then there's a metabolic step in the liver and another one in the kidney, and you get hydroxyl groups that are present at both of these moieties. It is, again, it's important. It's important at one level to know the words, the sequence of the words, but knowing that you gain breakage of ultraviolet at this base, and then you gain hydroxylation at this C1 and hydroxylation at C25. There are intermediate steps involved, redox reactions involving most likely adjacent carbons, and always consider the double bonds that you can get as to form the intermediate. That way you can understand it and not simply remember words in sequence. We talk about vitamin A now. Vitamin A is oxidized to metabolites retinoic acid and retinol, and these are involved in cell growth, development, and differentiation. All trans retinoic acid is metabolized from vitamin A. It's used in regulation. You can also use it medically for leukemia treatment and acne treatment. 11-cis-retinol conversion to all trans retinol is a reaction process in vision, and vitamin A can be stored in the liver as an ester with palmitic acid. Vitamin E is a class of lipids called tocopherols. These are antioxidants. They destroy free radicals, which would otherwise cause oxidative damage to cellular membranes. There's a rare deficiency in humans. For those who have a structured vitamin E, you can do rather elegant playing around with the free radicals that you can have vitamin E transform, but there is less of a biological utility, at least in studies involving vitamin E administration as a potential antioxidant in humans. Vitamin K is quite different. Vitamin K has a routine injection. Neonates that preclude hemorrhagic disease of the newborn. It is a factor in blood clotting. It facilitates conversion of prothrombin to form fibrin from precursor fibrinogen. For chemical analysis and characterization, you can extract lipids with ethyl ether, chloroform, or benzene, like nucleic acids, and unlike amino acids, these are rather hardy chemicals to work with. The membranes are more polar. They extract with alcohols. You can fractionate lipids with absorption chromatography, high performance liquid chromatography, thin layer chromatography. And you can use these for lipids with different polarities. If you have volatile lipid derivatives, you can additionally fractionate with gas chromatography. For chemical determination, you can hydrolyze all of the ester linkages using acids, alcohols, and enzymes. These are fairly straightforward identifications given the amount of repetition that is equivalent, and you could probably use mass spectroscopy quite nicely for this too, and in fact there it is. So this is a particular fingerprint that's utilized using these cleavage points that occur, and this gives a particular pattern that is characteristic of the fatty acid under consideration, these being the atomic mass units. Biological membranes and transport is the next major thing we want to look at. They have numerous functions. Again, it's a bilayer, and it allows for intermolecular collisions. So there's composition and architecture. The composition depends on the type of membrane studied. So in neurons, where you have a myelin sheath, these are lipid-rich, whereas the membrane that you see in mitochondria and in chloroplasts is protein-rich. Chemically, you can digest the membranes and disperse the components for isolation, and you will get a difference of membrane by site, difference of membrane between species with a spectrum of oligosaccharides, lipids, and proteins, and they will comprise the specific amounts that form the lipid membrane. The membranes are impermeable to polar solutes and permeable to nonpolar compounds. Again, the primary structural component is the fatty acid. And the fluid mosaic model that I referenced earlier, the nonpolar moieties, hydroxyls, carboxyls face the outer part, and the hydrophobic component, the breadth of the carbon, the methylene and alkene and alkane linkages face to the center. So the schematic is shown right here. You've got the outside and the inside of the cell, the bilayer here. Glycolipids can dot the surface. This is these are the type of cells these are the type of moieties you see in blood typing. You can also have oligosaccharide chains of glycoprotein and an integral protein with a transmembrane helix that occurs right here. This could be an alpha helix of protein. The lipid bilayer with the peripheral proteins that are on the center. You can have covalent links to lipids over here. Sphingolipids can be as numerous as the system requires, and polar heads are on the phospholipid here. So these membranes may look rather innocuous on light microscopy, but they are dynamic structures with a great deal of cellular activity. Some broad membrane principles to address. You have lipids. They form aggregates to create hydrophobic interactions. These will increase entropy and lower energy. The micelles are aggregates of lipids. These occur with free fatty acids and detergents. And the bilayer is unstable in an open form and would form a vesicle if there was fragmentation of the membrane. The components are synthesized in the endoplasmic reticulum. They're processed along the Golgi apparatus, and the composition within the Golgi apparatus changes during the processing. And then you can determine polar areas of lipids by attaching fluorescent labeling. Integral membrane proteins. These are proteins that exist between the two lipid monolayers. They traverse the breadth of the membrane. They're monotopic, where the integral protein involves one layer of the bilayer, and polytopic are proteins that involve both lipid layers. And some examples of ones that traverse both lipid layers are glycophorin that you see in erythrocytes, and oftentimes, as stated, the integral proteins restrict the hydrophobic component to the interior. Lipid bilayers are separated by about 30 angstroms, which imply about 20 to 25 hydrophobic amino acids in alpha helix, and that would span that distance. You can determine structural components with nuclear magnetic resonance and X-ray crystallography. We refer to a hydropathy index, which is the energy transfer that occurs when amino acids encounter water, which is higher for aliphatic and aromatic R groups, and then lower for polar groups. Easier to get that energy transfer when you've got a polar amino acid. Alpha helices line up more compactly given the structure of the alpha helix, and the beta sheets, which are in parallel open arrangement. These allow more for transmembrane transport. And some lipids are covalently attached to proteins. These are anchor proteins, and a lot of times you'll see these in the intestine. The intestine has specific transport mechanisms. They're often size and charge related, and oftentimes related to the type of food stuff that's involved. Membrane dynamics. The membrane exists in an ordered gel-like state, and the structural formations dictate its mobility. Some of these cells can change their membrane constituents in response to exogenous conditions. The transbilayer intracellular lipid movement usually requires energy and enzymes which can move lipid unidirectionally or bidirectionally, such as these terms are flipase, flopase, and scramblase, and these the prefix pretty much describes what the enzymes are doing to the lipid in the membrane. Lateral bilayer movement, where the lipids can trade places with adjacent lipids, so you can actually have movement of lipids along the membrane, and you can monitor this with fluorescent markers, and you can also see proteins migrating at different rates in the membrane. For specific interactions, we talk about sphingolipids and cholesterol. They form thicker macrodomains with less movement. They can move together through the fluid membrane, and in some instances can supply or comprise 50% of the membrane component. There is an integral membrane protein called calbiolin, and that creates infolding of a membrane, and this facilitates membrane recognition and transport, including in some components involved in signaling. So you get invagination of the membrane with infolding with this integral protein called calbiolin. So here's an example of that interaction right here. So you can see these invaginations here. This is a calvula right here, outside, inside, and this little vesicle that's forming, and the calvion dimer, which has 6 fatty acyl and 8 cholesterol moieties, occurs right here. So when you expand this part, this is what you're getting. This is a close-up of the structure, and then when you look at here's the calvule right here, and it can enfold, and then you can enlarge the cell by expanding the surface. So it has the ability to make the cell larger and smaller because of this invagination that occurs. So you can facilitate fusion of membranes of separate cells. This begins with increased curvature and protein scaffolding. So, for example, you can have a budding of vesicles from Golgi complexes. These are Golgi complexes. They bud off. You can have endocytosis, where material is brought into the cell. You can have viral infection, where the virus literally intercalates in this calvulin and then is brought into the cell. You can have fusion of sperm and egg. This is the same process occurring, modulated by calvulin. Fusion of small vacuoles that you see in plants, and separation of 2 plasma membranes at cell division. So all these are modulated by calvulin right here. All these endocytotic and processes of viral transmission and the like are modulated by this. And that will be it. Thank you very much.